Okay, well, today I want to try to do a quick lecture for you all on experimental design and experimental variables. As a part of today's discussion, I also want to look at this idea of a controlled experiment versus experimental controls. So we have kind of these three things that I want to look at with you, and I want to focus on something that is relevant right now, and that is the vaccine for COVID-19 and what's going on in terms of determining the efficacy of the vaccine. So I thought that this might be an interesting way to approach experimental design and experimental variables and experimental controls. So let's jump right into it. In previous discussions in class, we discussed the question and hypothesis generation for these vaccines. And I would just like to lay out a hypothesis. And the hypothesis might be something like this: a vaccine for COVID-19 will protect a sample of vaccinated mice more than those receiving a saline solution. And we could call this our hypothesis. I'm going to read it to you again. A vaccine for COVID-19. So that's assuming that we have some kind of chemical agent in there, will protect a sample of vaccinated mice more than those receiving a saline solution. We could also construct a null hypothesis, and our null hypothesis is the hypothesis of no effect, and that might say that a vaccine for COVID-19 will have the same effect on the mice as a saline solution. And so here what we are actually saying is that the vaccine and the saline solution will have the same exact response. It will protect the mice equally or fail to protect the mice equally. Neither one is better than the other. We would call this a hypothesis of no effect. Every time we do an experiment in this class, I am going to expect you to have a hypothesis that predicts the experimental variable will have an impact, and a hypothesis that predicts that there will be no effect or a null hypothesis. So I'm going to erase the board now. We're going to lay out our experiment and discuss with you how to control this experiment. So let's just erase this quickly. And we now have our sample. Now there are a few things that we need to do in our study. And I'm going to begin by saying that we need to control the variables. So I put up here on purpose some differences. You'll notice that I have a brown mouse and I have a white mouse, and they look different. You'll also notice that I have syringes with two different volumes of solutions. Now, we could ask the question, how does the vaccine impact type of mouse, and design the experiment for that? And in which case, we would want to compare brown mice to white mice. We could ask the question, does the volume influence how effective the vaccine is? And compare that. But in this case here, we are asking the question, does the vaccine protect mice more than a saline solution? And so given that that's our question, and our hypothesis is that the vaccine will protect mice more than the saline solution, or will protect mice equally, we need to control everything else. So for this experiment, I'm going to choose this mouse. And what's more, I'm not just going to choose this mouse, I'm going to choose this mouse times 100. So we want multiple individuals. And in a lab where they are actually constructing a vaccine, they often will use cloned mice, mice with the same genetic heritage, to make sure that the mice respond equally to whatever the test is. So here we are now controlling for mouse type. I'm going to continue to add to this list. Now the other thing we need to do is we need to decide on a sample size. And so I'm going to pick this syringe. And this syringe will have the vaccine in it. Oops, that was not what I wanted to do. So let me change that back. There we go. So this is vaccine. And we're going to vaccinate 100 mice with this vaccine. We're also going to take another 100 mice and we are going to fill it up with saline, or water, salt water solution. Now, I could pick either of my syringes, however, I actually need to keep the volume constant. So what I'm going to do is I'm going to pick this syringe, but we're going to inject it instead with saline. So we have the same type of mice, we have the same type of syringe, we have controlled for volume, we have controlled for syringe, we're going to try to control for the age of mice. We're going to control for the time of experiment. These are our controlled variables. In contrast, we would say that this is our experimental control because it tests for the null hypothesis. So that setup with the saline solution gives us something to compare to the vaccine. And that is our test for the null hypothesis. This is different from the controlled variables. Controlled variables are the things we keep same. These are the things we keep the same. Whereas the experimental control is used as a way to test if the null hypothesis is actually accurate. In contrast, we could describe this as the experiment with the experimental variable, where our experimental variable is the vaccine solution. So at this point, we should have a sense that there are two different things when we talk about controls. We can talk about control variables, which are things held constant. And our goal is to hold everything constant except for the experimental variable. We can also look at this as a controlled experimental design that allows us to test if our null hypothesis is true or if our experimental hypothesis is true. Finally, I'd like to remind you that every experiment must include independent variables where the independent variable could be thought of as three things. It is the experimental variable, the thing that you are testing, the thing that I change, or we could think of it as the manipulated variable. In this experiment, we are talking about the vaccine solution. Once we have determined that the vaccine actually works, if it does, we could then go to one of these controlled variables and design an experiment to see if the volume influences the vaccine. So controlled variables can become independent variables. The other type of variable is called the dependent variable. And this is your data collected. Here, our data collected is a comparison between the mice who are protected, so how many mice were protected by the vaccine, how many mice died, how many mice were protected by the saline, how many mice died. The dependent variable responds to the experimental variable. The dependent variable is the data we collect. Well, that's our lecture for today. I hope that all of this